Hey, hey, hey! I hope that you enjoyed your Christmas break. Sad it's over, me too. But I hope you're excited to start a new semester, PreAP biology. So go ahead and grab a highlighter, a pen, so you can take notes on the notes that are provided. So make sure you're writing down what I say to write down. Okay, so I think we finally reached the really good stuff of biology. Genetics. We already did before Christmas, we already did molecular genetics. And molecular genetics was like the DNA and the RNA and the genetics on a molecular side. So tiny, tiny, tiny. But now this unit, Unit 5, starts out with what we call Mendelian genetics, or like things you can see. Like I see that you have brown hair or blue eyes or a certain disorder. And so Mendelian genetics is genetics that is not molecular, more genetics that's big, like a trait or a characteristic. And the reason we call it Mendelian genetics is because of that guy right there, Gregor Mendel. And just looking at the history, 1850, this is a long time ago. He's considered to be the father of genetics. Now, he worked in a monastery, and he studied plants. So he was a gardener and a cook. But what he did was he studied pea plants, and he would cross them. And you probably remember something from middle school: the purple plants and the white plants, and such like that. And so Gregor Mendel is the person that studied the traits. And so you can see, looking at this cat, he has funny looking ears. But you can see he has, a certain color eye, a certain black coat. And so a character is an inheritable physical feature. So it says eye color or hair color there. But it could be something like height or like the texture of your hair. So anything very specific also. Okay, so a trait, this requires inheriting 2 alleles, one from each parent. Okay, so underline where it says 2 alleles, and right above that on the chromosomes. So when you look at a chromosome, like so, right? You've looked at a bunch of chromosomes already. There's an allele on those chromosomes. And you have one allele from your mom, let's say in orange, and we'll say in yellow, one allele from your dad. So you have 2 alleles, at least 2 alleles. Some things you have more than 2 for, but you have at least 2 alleles for each trait. You have 2. Now you always have 2, but there could be more than 2 possible alleles. Okay, so this is a variation of character, such as blue crossed eyes or black colored eyes, or black colored hair, sorry. Okay, so an allele is, and if we'll go to the next slide, here we go. An allele, and that's basically what I just drew. An allele is, underline, different versions of a gene. Different versions of a gene. Remember, a gene is a distinct DNA sequence that can make a protein. Okay, so if you look at a segment of DNA, a certain segment of DNA is a gene. Okay, and a gene has 2 alleles in it. Okay, so if you're looking at this, there's 2 alleles. That's the pink part. Here's your 2 alleles. 2 alleles for there. And together they make up the gene. So that's the gene for, looks like from the picture, that's going to be flower color. So this one is purple, and this one is white. Now one of those is going to be dominant, and one of them is going to be recessive. Okay, so a segment of DNA is a gene, and you have 2 alleles in every gene. Okay, so looking at your notes on B, it says each trait needs, underline again, 2 alleles. One from each parent. One from mom, one from dad. Okay, and those alleles are either going to be dominant or recessive. Okay, so the alleles are either going to be dominant or recessive. Dominant alleles are given a underlined capital letter. If a dominant allele is present, then that's what is shown. Okay, so if a dominant allele is present, then that's what you see. Okay, for example, the gene to be a dwarf is dominant. And so everybody that's a dwarf, everyone that has, and you know dwarfism is like short in stature, it's a genetic disorder. Everyone that has that disorder has at least one dominant allele. So me, I'm not a dwarf, so I have two recessive alleles. So if the dominant allele is present, then that's what you see. So let's say you have one allele to be a dwarf and one allele to be of normal height. Then you are a dwarf, because if the dominant gene is present, then that's what is expressed. Okay, recessive, given lowercase, so underline lowercase, and they're going to be covered up or hidden by recessive. So underneath recessive, I want you to write in for me masked by dominant gene. Or maybe under another thing to write under recessive, so also write this under recessive. Only shows up when dominant is not present. So the only way to express a recessive trait is if you have two recessive alleles. Okay, so the only way, you need to write that down too, I'm sorry. The only way to see a recessive trait is if you have two recessive alleles. Okay, so you must have two recessive alleles in order for it to show up. Must have two recessive alleles to show up. And I think you learned from my example, dominant or recessive, it's not that dominant is good and recessive is bad. That's not the case. Being a dwarf, that's a genetic disorder, which would not be a good characteristic. And so, in that case, dominant is bad. Okay, another example, sometimes it doesn't matter, good or bad. Brown hair, blonde hair, they're both great. So it's not necessarily good or bad. It's not necessarily color. But just some genes are dominant and other genes are recessive. So you want to for sure know those two words when you come into class tomorrow. Okay, so what Gregor Mendel did. Let's take a blank sheet of paper here. Okay, so what he did was he took a true breed. And you know what that is. Like, I have true bred or true breed pit bulls. And what that means is that their mommy is a pit bull and their daddy is a pit bull. And by the way, they're wonderful pets. They're not scary like you read on the news. Mine are soft and mushy and cuddly and wonderful, except sometimes they're not potty trained. Okay, anyway, true bred. You know what that means now, right? That means have a mom and dad and a grandparents that are the same type, right? Okay, so true bred. These organisms only have one type of allele. Okay, so for example, you have, let's go back there. You have two dominant alleles, or you have two recessive alleles. Okay, so if you have two, and you want to write all this in, write everything in that I'm writing down. If you have two dominant alleles, then we call that homozygous dominant. Okay, so homo means same, right? H-O-M-O, the prefix means same. Homozygous, these are not dominant, they are recessive. Okay, so be able to pick those out. Homozygous dominant is capital letter, capital letter. Homozygous recessive is, homozygous dominant is capital, capital. Homozygous recessive is little case letter, little case letter. Okay, so if an organism has a capital letter and a lowercase letter, then they're not homozygous. They are, think back to the prefix and suffix game, they are heterozygous, right? Okay, so you want to know all three of these vocabulary words tomorrow. Homozygous recessive, homozygous dominant, and heterozygous. Okay, so take a second, pause the video, get all three of those words down. Homozygous recessive, homozygous dominant, and heterozygous. And, you know, it doesn't matter what letter it is. Like, it could be an S or a B or an A or, you know, it doesn't matter what letter. Just capital, capital, lowercase, lowercase. Okay, so back to Gregor Mendel. This is perfect because I can use purple. He took a purebred purple flower. And for the picture, we'll draw black being white. So a purebred white flower. Okay, so this is what he called the parent generation. Okay, and they are, like your notes tell you, pure or true breed. True breed. There we go. Okay, and he crossed them together. What that means is he made them make a baby. He took the pollen of, let's say, this one, and sprinkled it on the white one. So you remember from elementary school, he crossbred them. So he took the pollen here from this flower. Look how cute it is. And he put it over there. Right? Okay, so let's get back to the story. Okay, so Gregor Mendel took the purple purebred and the white purebred, and he mixed them together, and he crossbred them, and this is what he got. Who knows? What color? Now, he didn't just have 4 babies like I'm about to draw, but it represents a percentage. So, like 25%, 25%, 25%, 25%. Lo and behold, a purple. 25% of them were purple. 25% of them were purple. 25% of them were purple. All right. I want you to think for a second. Think back to 7th grade. Was 25% of them white, or were 25% of them purple? And the answer is, ding, 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 purple. Okay, so when you mix a purebred flower and a purebred white flower, you get 100% of the time purple flowers. So you know that put yourself in Gregor Mendel's shoes. You're sitting there in your garden in 1850, a long time ago. No iPhones yet. Sitting there in your garden, and you mix a purple flower and a white flower, and all the baby flowers are purple. In his head, the question is the white still there? Somewhere inside, oops, I wanted that to be gray. Somewhere inside that purple flower, is there white? I just can't see it. Now, it's not like a light purple. It is purple, 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 like the exact same color as that purple. It's not lilac or whatever color that is, like light purple. It's like really purple, dark purple. Okay, something I want to back up a little bit. How did he know, because he didn't have like microscopes or DNA or anything like that, how did he know that that was purebred? It's because he mixed it like a purple flower, made purple flower. If it self-pollinated, all it made was purple. Like the only DNA inside that plant is purple. How did he know that was white? All pure white. And that's because if it made babies by itself, because you know plants can self-pollinate, it made all white. It's mom and dad, per se. Plants aren't girls and boys, but both parents were white. Okay, so next he was like, well, if that makes all purple, what happens if I mix these two, or any two of the, we're going to call this the F1 generation. So that, it stands for filial, the F1 generation. Okay, so we mixed them together, and we need 4 of those little things. What was the outcome? Do you know? You know we got at least one. You mix a purple and a purple. Well, you got some purple. 25% at least are purple. Well, that makes 50% purple. 75% purple. Oh, but wait a minute. 25% show, so 25% show the recessive trait white. This is 75%. Hope you can see that down at the bottom. Okay, so 25% are white, 75% are purple. And this is the F2 generation. Okay, so why? Why did that? Okay, so here's Gregor Mendel. You mix purple and white, you get all purple. Okay? So if you mix purple and purple, you get 75% purple, 25% white. What? Okay, let's talk about the science behind this. This, what you see, is the phenotype. Look at your notes where it says phenotype. Phenotype is the physical appearance. You want to know that word tomorrow, so make sure you got that one. The physical appearance of something. I have brown hair. I have blue eyes. I am 5'7". That's my phenotype. Okay, my genotype is going to be my DNA, my genetic makeup. This is something you can't see. You can't see my genotype unless I'm homozygous recessive. Then I show the recessive trait, like I'm not a dwarf, so you can see you can't physically see it, but you can know my genotype. Okay, so let's look at the science behind this. If you cross the purple, this first one is going to be, we'll use the letter A, big A, and then the recessive one is little a, little a. Okay, when you cross those together, you got 100% big A, little a. So that's what these are. They are heterozygous. So I'm going to, let's see. Okay, so you got your big A, little a. No, sorry. Big A. Crossed with little a, little a. So this is like purple and white. Remember, this one's purple. That one's white. Cross them, and you got 100% big A, little a. Okay, let's talk about why. Look at the picture, and you got one from mom and one from dad. So you got a capital letter was given to all 4 offspring, or all 100% of the offspring. A lowercase letter was given here. And this looks confusing. So scientists made up something called, you know this, a Punnett square, to make it really easy to do. And I know you remember that from 7th grade. But then when you cross these two, so I'm just going to show you the genotypes here. When you cross these two, you got 1 white and 3 purple. Do you see how this is purple and this is purple? And this we know is white, because 2 recessive traits makes the recessive allele show up. This heterozygous has the recessive traits, but it doesn't show up. Okay, so and hybridization would be heterozygous. So do you see that in your notes? Heterozygous hybridization. Okay, and you got genotype and phenotype. Let's move down in our notes to Punnett square. Okay, so when you're looking at the notes, this is what Gregor Mendel did first. Punnett square, he crossed homozygous dominant with homozygous recessive, and then you bring the capital letter down and over, like that. So capital letter down, little case letter over. Okay, so let's see if I can draw this a little bit better. The letters on the top are always going to go down into the block. The letters on the side are always going to go over. Okay, so let's try another one. Let's see if you can do it real quick without me, and then I'll tell you the answer. Big A, little a, big A, little a. This is what he did with that F2 generation. Okay, so pause the video and see if you can fill that out. I want to see these Punnett squares on the back of your notes. So turn your notes over and draw this. See if you can fill it out. Pause the video. So if that's what you got, you did it right. Okay, I also want you to be able to see that this one is recessive. Homozygous recessive. These two are heterozygous. And then that one is homozygous dominant. Okay, so take a minute and see if you can do that. Homozygous dominant, 2 heterozygous, and 1 homozygous recessive. All right, let's try one more and we'll move on from this. And this is a monohybrid cross, by the way, because we're only looking at one trait. So big A, little a, little a, little a, go. See if you can do that one. Pause the video. All right, and if you got that, you're right. Okay, what percent is homozygous recessive? What percent is heterozygous? Okay, what percent would be purple? 50. What percent would be white? 50. Good. Hopefully you got that. Okay, so that's a monohybrid cross because you're looking at one trait. And we're going to practice some more of those in class tomorrow. A dihybrid cross, you know this, you're looking at, underline the word, 2 traits. Trihybrid, you're looking at 3 traits. And those get pretty confusing, but I'm going to teach you a shortcut in class. Okay, Mendel, from looking at these pea plants, and let's exit this and go look. Okay, so here's your dihybrid cross, and you can see the peas that Mendel studied, they had more traits than just color. Like I was talking about the flower color, but they had seeds that were either green seeds or yellow seeds. Okay, and then of those seeds, they were either round, which you can see in this yellow one right here, or wrinkled. So the seeds could be green and round or green and wrinkled. Yellow and round or yellow and wrinkled. And so just like you can be tall with brown hair or short with brown hair. And your hair color has nothing to do with your height. You know, you can have your mom's height but your dad's hair color. Or vice versa. You know, those two don't have anything to do with one another. Okay, so Mendel came up, by looking at all the things that he studied, he came up with 2 big laws. So you want to know these 2 laws tomorrow. Law of segregation and law of independent assortment. Okay, first one. The law of segregation says that alleles on the homologous chromosomes, alleles on the one from mommy, one from daddy, homologous chromosomes, move, circle the word, independently of one another. They walk to opposite poles of the germ cell, sex cell, right above germ cell, sex cell, sperm or egg. During anaphase, this separation is continued through anaphase 2 when the sister chromosomes are isolated individual gametes. This occurs at anaphase 1 and 2 in meiosis. Okay, that sounds confusing. Let me tell you what it means. It's really way easier than it sounds. In meiosis, and that's how you make sex cells. We kind of touched that last semester, but we will touch it some more. The chromosomes line up in metaphase, and then, look, the chromosomes separate. So you could get chromosome number 1 from your mom, and chromosome number 1 from your dad, but you could get both of mom's or both of dad's, like they separate independently. So the first law is just saying that they separate. So those chromosomes separate in anaphase. Remember from mitosis how they separate? Well, they separate the same way in meiosis. And then the second law of independent assortment, here it shows this law describes the behavior of the chromosomes in metaphase 1. So really what it is that they line up And they can line up, if you look, like they can line up blue on the left, red on the right, or vice versa. So they line up and they separate, is what the first one says. And the second one, independent assortment, says that they separate independently of one another. So they separate independently. So the chromosomes line up and they separate. But they separate regardless of the other one. So, like I said, hair color has nothing to do with eye color, but it does a little bit. Hair color has nothing to do with your height. So that's an example of independent assortment and segregation, really. Okay, and then the last thing on the notes is probability. You already know this. If you flip a penny, you got heads 50% of the time, tails 50% of the time. If you flip it 100 times, chances are you're going to get about 50-50. You know, maybe 49-51, something like that. Okay, so let's say you flip a penny and it lands on heads. That's the probability, 50%. What if you flip a penny? Okay, so one thing to know about probability, if you flip a coin 10 times, and then you're about to flip it on the 11th time, you still have a 50-50 chance. Just because you have heads 3 times in a row, it's still a 50-50 chance that it'll be tails the next time. So it's still half heads and half tails on the penny. Okay, so probability refers to the likelihood of a certain outcome happening. Probability ranges in a scale from between 0 and 1%, or 1 or 100%. On a monohybrid Punnett square, each square represents 25%. So we talked about that. When you look at the Punnett square, it's not like every flower has 4 babies. No. It's 25% of the time, it's going to be this, and this, which equal up to 100%. And you add all the boxes that have the same genotype. Remember, that's the DNA that you can't see. And that's how you get the probability. Okay, so tomorrow, when you come in class, make sure you know these words. Dominant. So write these down on the back of your paper. Make sure you know them. Recessive. Homozygous dominant. Homozygous recessive. Heterozygous. Phenotype. Genotype. Law of segregation. Law of independent assortment. Okay, so tomorrow you got 1, 2, 3, 4, 5, 6, 7, 8, 9 things to know. I hope that this video was helpful.